I am Maryam Malik from Rawalpindi Medical College. The topic today is mycology. We will begin with the differences between the fungi and bacteria. This is table 47-1 Levinson. The main differences between fungi and bacteria are: the fungi are eukaryotic and bacteria are prokaryotic. In the cytoplasm of fungi, mitochondria and endoplasmic reticulum are present. In case of bacteria, the mitochondria and endoplasmic reticulum are absent. The cell membrane of fungi contains sterols. The sterols are absent in bacteria, with an exception to mycoplasma. The cell wall of fungi consists of chitin, and the cell wall of bacteria consists of peptidoglycan. There are two types of fungi: the yeast form and the mold form. The yeast grow as single cells that reproduce by asexual budding. The molds grow as long filaments that are also called hyphae. They form a mat that is called mycelium. Some hyphae form transverse walls called septate hyphae, and some do not form. They are called non-septate hyphae. When there is no septum in between, there will be many nuclei, so the non-septate hyphae are also called cenocytic hyphae or multinucleated hyphae. The fungi can be monomorphic or they can be dimorphic. Dimorphic means they form different structures at different temperatures. They exist as molds in the environment at ambient temperatures and as yeasts in the human tissues at body temperatures. Fungi can reproduce by either sexual reproduction by the help of sexual spores or asexual reproduction by the help of asexual spores. The sexual spores are zygospores, ascospores, and basidiospores. These fungi are called perfect fungi. Those fungi that do not form sexual spores are termed imperfect fungi. They reproduce by the help of asexual spores that are called conidia. Some important conidia are arthrospores, that are the mode of transmission of cocoid, chlamydiospores, that are present in Candida albicans, blastospores, sporangiospores, they are present in Rhizopus and Mucor. Now the classification. The fungi are classified into superficial mycosis, cutaneous mycosis, subcutaneous mycosis, systemic mycosis, and opportunistic mycosis. Some of the books regard superficial mycosis as a part of cutaneous mycosis. Superficial mycosis. The fungi is Malassezia furfur, and it forms a rash that consists of multiple hyperpigmented and hypopigmented areas. This is termed as tinea versicolor. The second is Exophilia wernicki. It forms dark patches on different areas of the body, and this type of rash is called tinea nigra. The superficial mycosis can be diagnosed if we treat the skin scrapings with potassium hydroxide. This shows spherical yeasts and branched hyphae. These are the branched hyphae, and these smaller ones are the spherical yeast, like the bunch of grapes. The treatment is by the help of selenium sulfide shampoo and various imidazoles. Cutaneous mycosis. Cutaneous mycosis is done by three important fungi: epidermophyton, trichophyton, and microsporum. These three cause dermatophytosis. They are also called dermatophytes. They can do tinea corporis, involving the whole body, tinea cruris, tinea pedis, tinea capitis, involving the head, tinea unguium, involving the nails, tinea barbi, involving the beard. This is the picture of tinea corporis. It is commonly called ringworm. This is the picture of tinea pedis. It is commonly called athlete's foot disease. The cutaneous mycosis can be diagnosed with the help of potassium hydroxide. In cases of some species of microsporum, Wood's light is used. In cases of cutaneous mycosis, no yeast is seen. We can see branched hyphae. These are the branched hyphae that have formed a mycelium. And these are macroconidia. Since the microconidia are very small, they are not properly visible over here. The treatment is topical imidazole, and if there is the involvement of hair and nails, then we have to give griseofulvin. In the next section, we will discuss the other types of mycosis. Thank you.